Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. So in this video we will talk about the stomata function, that's the physiology and anatomy. So first of all I will give you a concept about the before the understanding of the stomatal opening and closing. So here is the plant which that is contain a shoot system and root system. There's the leaf and stem is here. There's the basically root which that are used for the absorbing of nutrients, and stem and leaf is used for the erection and retaining, and leaf is used for the photosynthesis in the form of light absorption to produce a byproduct is oxygen and water vapors. And after the evaporation and water vapors and oxygen will be released, and in the other hand is the sugar will be produced. So now we will talk about the cross section of the leaf. So here I draw a leaf which that is, here is the midrib and there's the vein, and there's the leaf is the basically simple leaf which that is cross section of the tip of the leaf we will draw here. So the leaf contain a mesophyll cell. First of all I will say that the here is a leaf, and we will discuss about the here is the epidermal layer, and there's the epidermal layer of the basically upper epidermis, and here is the mesophyll cell, and here is the basically midrib. There's a midrib contain vascular bundle, and is the down is the hypodermal or lower epidermis layer. So the lower epidermis layer contains stomata. There's the stomata is function to basically here is the midrib which contains xylem and phloem as a midrib, and here is the xylem will excrete water from the root absorption and go into the air space is also present, and palisade and spongy mesophyll cell are present. So now we will talk about the basically the stomata which that are used during the daytime for release oxygen and water vapors is in the byproduct of the photosynthesis. So it means the byproduct of photosynthesis is the basically oxygen and glucose. There's the basically production due to the photosynthesis. There's the chlorophyll are present in the palisade mesophyll cell and spongy mesophyll cell. That's a use for the chloroplast which that we will discuss about. And here is the low level of carbon dioxide is responsible for the open stomata and increase oxygen in the cell of leaves also. And here is the stomata which that open during the requirement of intake of the carbon dioxide or release of the water and the release of the oxygen. So here I draw a palisade mesophyll cell which that are structure is here nucleus and vacuole are present, and the important organelle which that are chloroplast. That chloroplast is used for the basically photosynthesis. So the chloroplast is used for the photosynthesis is here. I'll draw a chloroplast which that contain double membrane organelle. There's a double membrane organelle contain thylakoid. There's a thylakoid membrane is contain the grana. There's the single is granum and multi grana, and the stroma is present between the thylakoid, and there's the here the light will disperse and light will enter into the chloroplast, and there's the light in the presence of chlorophyll. That's the pigment are present in the thylakoid membrane which that convert oxygen is the byproduct, and water vapor in the form of water, that's and water molecule and sugar. That is the two hypotheses which that are we will discuss is now here about the stomata opening. So the stomata is made up of guard cell. There's a two guard cell in the form of kidney shape. So here the starch sugar hypothesis we will discuss first. There's the German botanist H. van Moel proposed that. So here the sugar are accumulation during daytime, and that means the sugar will accumulate into the guard cell. That's the solution into the guard cell is concentrated solute. That's a concentrated solution will is the responsible for the entering of water into the guard cell. That's the entering of water into the guard cell is the responsible for the turgidity. So I hope you make sense that the aquaporine channel are present in the guard cell which that are water absorbed into the guard cell due to the osmosis, because that the guard cell contain too much sugar, and that's why that's proposed about this. But in the other hand, we will talk about the potassium influx hypothesis. In the pharma potassium influx hypothesis, according to this, the potassium ion will enter into the guard cell, and hydrogen in the case of outward in the that is the active transport, and ATP used for the production of basically going in inward the potassium. That means the inward moving of the potassium will increase the basically solute and concentration. So it means the solution concentrated solution will be influx the potassium is give the concentrated solute, and water potential will be low and water move inside. That means the water move inside, and basically when water move inside the automatically you know about that the stomata will be guard cell will be turgid, and the stome which that are present in the pore which that are open. And now we will talk about the advancement. So advancement means the mesophyll cell which that are present in the upper side of the stomata, basically guard cell. And here is the stress hormone which that are phytohormone. When stress will be occur drought or some stress with that high intensity light which that are abscisic acid, jasmonic acid, and brasinosteroid, cytokine, ethylene which are responsible for the entering into the guard cell, and abscisic acid is responsible for the release of the water from guard cell and shrinking the guard cell to close the guard cell. And in other hand, when ABA and jasmonic acid, nitrite and nitric oxide, which is responsible for the collection of this hormone, is the closure, and auxin plus cytokine is responsible for the opening. So it mean is the closure due to the ABA jasmonic acid plus nitric oxide. So in other hand, the auxin plus cytokine kinin which that are used for the opening. So now we will discuss about the method of the chloroplast opening and closing. So here first of all we will discuss about the opening. So now we will go into the closing. So here I write closing, but it's not in this closing, that we will discuss in the next. So here is the first of all we will discuss about the opening. There's a phytohormone, abscisic acid, and plus another hormone which that are not allowed to enter into the guard cell. So it means as abscisic acid which that are already present in the guard cell which that also catabolized, and too much decrease of the abscisic acid which responsible for the entering. Due to the decreased abscisic acid, hydrogen proton and chloride will enter into the channel from channel, and potassium inside move, and cat channel are present, and ATP used for the ATP, that's the active transport. And here is the nitric acid which enter by the one proton inside moving, also responsible due to the basically chlorina one channel. So here is the basically another important ion, malate ion to negative which that are enter, and starch synthesis that's known as glucogeno genesis, gluconeogenesis, which that is increase the glucose and accumulation of too much solution into the guard cell, which that are responsible for the entering of the water. So now we will talk about the basically closing of the basically we will talk about the closing of the stomata due to the guard cell. So here is the basically synthesis of abscisic acid and jasminic acid will enter into the guard cell, which that are responsible for the chloride ion S type channel, which that are excrete, and nitric oxide, which that are R type channel, which that are excrete, and potassium will excrete due to the this entering of the hormone. So malate ion also excrete. It means the starch will not convert into the glucose, and calcium ion go inside for the actin protein, which that are used for the constriction. So calcium is very important for the so here is the abscisic acid, which that are abscisic acid, which that is basically RPP2, which that are go into the SNRK, which that are the changing of the protein, which that abscisic acid that is responsible for the activation of gene, which that activation of gene which that are triggered to excrete the water from the guard cell. So excretion of the guard cell is the aquaporin will release and chloride ion will be released also. So that's the whole process is in the uh, formation about the closing, and uh, here is the aquaporin and closing and opening of the stomata due to the chemical reaction and the chemical changing. So here is the basically I will mention the removing of water decrease turgidity of the